So welcome back. Now we will listen to the third talk of this conference, and it's given by Boris Schmidtgall. And Boris, he started studying chemistry at Hamburg University, and then he took a PhD at the University of Göttingen, Germany. And his thesis was about modified DNA molecules. And Boris Schmidtgall has also studied some the chemical properties of some flexible proteins. And he's also chairman of the German creationist organization, Nordic, which I think is the largest creationist organization in Europe. And we're very pleased that Boris will give the next talk here, and the title is Review Creative Diversity in Life's Light Harvesting Systems. So welcome, Boris.
So, thank you, Ola, for the kind introduction, and greetings to all of you. Yes, I studied chemistry, and in a similar way as Peter, I converted to Christian belief because, one of the thoughts that led me to the conversion was that nature does not function in the way evolution presents it. And I observed simply observed chemical reactions and laws, and came to the conclusion that creation is more plausible than evolution. And during my studying time, I was focused on large biomolecules such as DNA and proteins. I synthesized them, and I observed their properties, and this is also what I more or less do now. I focus on the chemistry and biochemistry of the cell, and I'm not, just to correct, I'm not the chairman, I'm a CEO. So this is a difference. The chairman of Wort und Wissen is Hendrik Ulrich, someone else than me. Okay, and my today's talk is Creative Diversity in Life's Light Harvesting Systems. And this is a very interesting thing, because this is the base of what life on Earth is. Everything depends on light on Earth. And there is also a parallel to the Bible. The first thing that God created, according to the creation account, was light. You can see this in Genesis chapter 1, verses 2 and 3. There is written, "And the earth was without form and void, and darkness was upon the face of the deep. And the Spirit of God moved upon the face of the waters, and God said, Let there be light, and there was light." So, for all we know, it was not the physical light, we know, as the sun was not created until the fourth day. Therefore, the first organisms saw the light of God and not the light of the sun. And there is no question that we are more dependent on the still invisible light of God than on the visible light of the sun. It is said that the Lord and Savior Jesus Christ is the light. In John chapter 1, verses 4, it is stated, "In him was life, and the life was the light of men." So we must receive the word of God in order to live. And this is a kind of, that we can do that because God has endowed us with the appropriate perception. And in a way, creation is a parable for us that shows our dependence on God. Almost all life depends on light as its primary source of energy. Light is undoubtedly the largest source of energy available on our planet. But for light to be utilized by organisms, they must have highly efficient energy conversion devices. So this is the parallel. God has endowed us with the capability of understanding His Word, of seeing His invisible light. And God has endowed life with sophisticated molecular mechanisms in order to harness natural light. Nature must harvest light in order to live. And to do this, it has been equipped with complex molecular systems that enable it to do so. And today I would like to introduce the light harvesting systems that exist in nature and to show why they are a powerful testimony to God's creation. So, first of all, organisms have different ways to transform energy. So all organisms depend on some kind of nurture. So there is this Greek word that is used, trophic. So they feed themselves somehow. For example, us, we are heterotrophic. That means we consume organic matter. And then there are some organisms that consume minerals. So they eat stones. They are lithotrophic. And today I'm going to talk about phototrophic organisms solely. I'm not going to mention the other ones. And the point is here that the energy obtained from the nurture is converted to energy currencies such as ATP and NADH. And this energy is then used for vital functions such as perception, motion, metabolism, thermal regulation, and so on. So, when classifying organisms, the source of carbon is often also mentioned. For example, organisms that obtain their energy from light and their carbon from carbon dioxide are called photoautotrophs. If carbon is obtained from organic compounds, we speak of heterotrophic organisms. So, but I'm going to talk only about energy conversion and not about the source of carbon. All phototrophic organisms make use of photosynthesis apparatuses. That means ensembles of protein complexes that enable the conversion of light energy into chemical energy equivalents. So photosynthesis is defined as a process in which an organism catches and stores light energy and uses the stored energy to drive energy-intensive cellular processes. And plants are a typical representative of photosynthesizing organisms. This is the commonly known type of photosynthesis. So light, water, and carbon dioxide are consumed, and oxygen and sugar are produced, as we can see in this figure here. This type of photosynthesis is called oxygenic because it produces oxygen. The process described takes place in chloroplasts. These are cell organelles that contain densely stacked thylakoids. So the thylakoids, we can see them here in the picture. Here, these are the thylakoids. They are containing many copies of molecular machines that store light energy in the form of chemical energy equivalents. So there are also bacteria, not only plants, but also bacteria that can utilize light energy. The only bacterial strain that carries out oxygenic photosynthesis is cyanobacteria. These are prokaryotes that have thylakoids as organelles. And they convert light energy into energy-rich chemical compounds. So then we have also some organelles. For example, the thylakoid, this is an antenna molecule, or this is an antenna complex, which mostly contains chromophores, so that are molecules that absorb light. I will describe them later on. And then there are some other organelles, for example, here the carboxysome, which contains the Rubisco enzyme and the carbonic anhydrase. In this carboxysome, the fixation of carbon dioxide takes place. So as we can see, cyanobacteria are already quite complex organisms. And they form such chains, such fibers in water. This is a photograph where it has been enlarged. And when it gets hot in the summer, a lake can bloom because of such cultures. Yeah? When they grow quite quickly, then the lake can bloom in such a way. So cyanobacteria are also oxygenic. But most organisms actually that perform photosynthesis are not oxygenic, they are anoxygenic. That means they also receive light and harness it to produce energy equivalents, but they do not produce oxygen. And mainly, these are exclusively prokaryotes, such as purple bacteria, green sulfur bacteria, non-sulfur bacteria, heliobacteria, chloroacidobacteria. They are quite different in the way they perform their energy metabolism, and they all perform anoxygenic photosynthesis. Then there is a third type of photosynthesis. It is the rhodopsin photosynthesis. These are mostly extremophilic archaea. So, for example, halophils or thermophil microorganisms. So the basic principle of photosynthesis is depicted here in this scheme. This is, so to say, the heart of the photosynthesis, because it is this the unit where the storage of energy takes place. So the energy is extracted from light. Light causes the excitation of an electron, and the electron then is the electrons they form a current, and the current ends up at the NADPH. And at the same time, this light drives the pump, which pumps protons from one side to the other of the membrane, thus creating a gradient, and this gradient drives the ATP synthase. So in this way, excitonic energy, which is prone to rapid heat dissipation, is converted into a more stable form of energy and stored for further applications. And the main components of photosynthesis are the proton and electron pump, which derives the electrons. In the case of anoxygenic photosynthesis, the electrons are derived from ions, hydrogen, sulfur, or sulfur hydrogen, And in the case of oxygenic photosynthesis, the electrons are withdrawn from water. So this is the basic principle of photosynthesis, and there are also some challenges to photosynthesis. First of all, the energy that is obtained by such organisms comes from light, and light is a diluted energy source. So it arrives at the Earth at a limited spectrum, and the usable spectrum for photosynthesis apparatuses is 380 to 1000 nanometers. So this is the wavelength of the radiation that is used by photosynthesis apparatuses. And light of short wavelengths is damaging to the photosynthesis apparatus, whereas light of longer wavelengths than 1000 nanometers transfers too few energy to drive photosynthesis. And then, of course, there is also the light has a different quality and intensity. For example, on the surface on the water, there are mostly organisms that absorb light in the red and the blue region. And some meters below the water surface, there are mostly organisms that absorb light in the region of the green wavelengths. Yeah. And then there is a champion, the green sulfur bacteria. They consume light at a depth of 80 meters below the water surface. So these organisms thrive at a depth of 80 meters, and they are autotrophic. They are photoautotrophic. And they receive 300 photons per second. So this is very few. Just to compare it to plants, plants receive 10 to the power of 16 photons per second, a normal plant. It's a huge difference. So there are enormous differences between the conditions under which the organisms live. So photosynthesis has to be highly various. Yeah? It has to be endowed with very different molecules to function in very different contexts. And one of the important aspects, it is called, so to say, the blood of photosynthesis. It is maybe the most important component of photosynthesis apparatus. These are the chromophores. The chromophores are large molecules that are also quite hard to synthesize. So, for example, this chlorophyll has been synthesized only in the year 1965, and the one who synthesized it received a Nobel Prize for it. So this is really a work of art to synthesize such molecules. And phototrophic organisms have very differently, or have slightly differently designed devices for concentrating light energy. And the devices are called light harvesting complexes. They are coated around the reaction center of photosystems, and they function as a kind of energy funnel to concentrate light in the reaction center. The interesting point about such molecules, the chlorophylls, is that their extinction coefficient is very large. It is around 100,000 per mole per centimeter. So this is the largest number known for such rather small molecules. And then there are other chromophores, for example, such as carotenoids, and also open-chain tetrapyroles. And they vary in their absorption maximum, so that light in different absorption maxima can be absorbed. At the same time, carotenoids function also as scavengers of radicals, so they are also important for the so-called photoprotection of photosystems. Another important aspect is the metal, which is coordinated in this ring system. It is magnesium. So in magnesium is rather unusual to be complexed by tetrapyroles, except for those used in photosynthetic apparatuses. In many other such molecules, so in many other tetrapyroles, iron is complexed, but not magnesium. And the point here is that the magnesium ion is ideally suited to stabilize excited singlet states. So excited singlet states have to be transferred from the edge of the light harvesting system to the center. And if there is a strong spin-orbit coupling, for example, with iron, there is a much stronger orbit coupling, and this does not allow the excited states to be stabilized, whereas magnesium allows this. So magnesium kinetically inhibits the transition to less energetic states, to triplet states. It is mainly for this reason that the lifetime of excited states of chlorophylls is significantly longer, roughly some nanoseconds, than the process of excitation, which is about 20 to 300 picoseconds, depending on the photosynthesis apparatus. And then there are some acidophilic phototrophic bacteria that contain zinc instead of magnesium. This also exists. The reason for this is that in strongly acidic medium, magnesium would be replaced by protons, which does not happen when there is a zinc ion. So this is a scheme of a photosystem. So I call those proteins, which contain the reaction center in the middle, I call them photosystems. And then there is this light harvesting complex wrapped around the photosystem. And what it does is it transfers energy from the edge to the middle, to the center, and then excitation happens. So, interesting point about this is that the absorption maximum of the chromophores here is different. For example, here in the outer region, the absorption maximum is lower. It means light of shorter wavelengths is absorbed in the edge. And wider, and in the region 2 or 3, the absorption maximum is higher, so the wavelengths are longer. Why is that so? This arrangement is necessary for the energy to be concentrated in the center. So this is a directed flow of energy from the edge to the center. And at each transfer from chlorophyll molecule to chlorophyll molecule, there is a small loss of energy, so that this transfer becomes irreversible. And by becoming irreversible, it cannot return. So this is a kind of energy funnel. And it is also called a coherent excitation transfer. And the number of pigment molecules in light harvesting complexes is highly variable in organisms, ranging from a few tens to over 1,000. Just imagine this, the bacterium thriving 80 meters below the water surface. It will have quite large light harvesting complexes, whereas a plant does not require such large light harvesting complexes because they can also be damaged and they have to be repaired. So the decisive event, meaning the excitation of an electron here in the reaction center, is enhanced very strongly by the light harvesting complexes which are wrapped around the photosystem. It is approximately a factor of 100 that this process is enhanced in comparison to the situation when the light harvesting complex was absent. So in this step, the excitation of an electron is also called charge separation, as I will show later on. So obviously, this whole thing is a highly teleological structure, the origin of which by no means can be well explained by evolution. As you can see, it is created to concentrate light in the center. Yeah, and there are about 100 chromophores arranged in a highly ordered manner to perform a coherent energy transfer from the edge to the center. However, light as an energy source has also a downside. It causes the formation of reactive species, such as singlet oxygen or various radicals, such as superoxide, peroxide, or the hydroxyl radical. And reactive oxygen species are formed constantly and in many ways whenever oxygen comes into contact with, for example, metal ions such as iron 2 ion or copper 1, organic molecules, and more than 40 kinds of enzymes. So there is a necessity to constantly protect the vulnerable components of the photosynthesis apparatus against photo damage. And Blankenship comments on this with the following words: antenna systems as well as reaction centers therefore have extensive and multifunctional regulation, protection and repair mechanisms. So, for example, there are such molecules as vitamin C. Vitamin C in plants is imported into the chloroplasts in large amounts. This is done because chloroplasts are vulnerable against reactive oxygen species, and they have to be protected. Vitamin C is a scavenger. And then there is, for example, there are such molecules, they are called phenols or polyphenols, such as resveratrol. And such molecules also act as scavengers of reactive oxygen species. So this phenomenon is called photoprotection. Without photoprotection, photosynthesis apparatuses would be degraded quite quickly. Small molecules, small organic molecules are required to scavenge reactive oxygen species. Then there are complex enzymes that are required to constantly repair the photosynthesis apparatus, and there have to be also detection systems, because there are some molecules that enhance the formation of reactive oxygen species. And there are detection systems that help identifying them and that signal to produce more photoprotection devices. So there are three main types, as I already mentioned, there are three main types of photosynthetic apparatuses in nature. There are two kinds belonging to the chlorophyll photosynthesis. So they are defined by the components that compose their photosystems. And the oxygenic and the anoxygenic photosynthesis that belongs to the chlorophyll photosynthesis is quite different. And then there is a third one. This is the rhodopsin photosynthesis, and this again differs much more even from the other two. In chlorophyll-based photosynthesis apparatuses, the energy of the photon is transferred to electrons, resulting in a flow of electrons along the membrane of the corresponding organelle, which drives proton pumps and thus enables the buildup of a transmembrane potential. This potential is utilized by ATP synthase to store the energy in the form of ATP, and the electrons are stored in energy-rich molecules. This means reduction equivalents such as NADPH. Chlorophyll-based photosynthesis apparatuses can be further subdivided, as I already mentioned, in two groups. So the oxygenic and anoxygenic. And one of the main differences also is the oxygenic photosynthesis apparatus contains oxygen evolving complex, which is seen here. So, and the anoxygenic photosynthesis apparatus has a cyclic electron transport, whereas the electron transport here is linear. This already shows the basic differences between these kinds of photosynthesis apparatuses in nature. So let's see some details. Here I omitted the light harvesting complexes that are coated around the photosystems for clarity. And this is the most complex photosynthesis apparatus called oxygenic photosynthesis. So it is composed of 5 components. We have the photosystem 2, photosystem 1, the BC1 complex, and the ATP synthase. And the electron flow starts in the oxygen evolving complex. There, water is oxidized, and this is a chemically very demanding process. So water is actually not a molecule that is easily oxidized, but in the oxygen evolving complex, this happens easily. And the electron is then transferred to the photosystem 2, and the electron flow goes via the photosystem 2 and then BC1 complex. And here, a very interesting thing happens. The electron flow is bifurcated. So electron bifurcation occurs here in this BC1 complex. It is a fascinating process that has been studied in depth only in recent times. The first ones to discover the bifurcation were researchers in the 1970s, but until today many things were not well understood. So this is a chemically very demanding and complex process that serves to increase the energy yield and is also only possible thanks to very fast quantum mechanical tunneling processes that require a precise arrangement of cofactors with exactly fitting photochemical parameters. So as we can see here, 2 electrons arrive here from complex 2. The complex 2 is the photosystem, or so to say, here's the quinone pool. And quinones are molecules that transfer 2 electrons. But here in the plastocyanin, only 1 electron can be transferred. So the problem that is solved here is the switching from 2 electron transport to 1 electron transport. And this is why the bifurcation is necessary in this complex 3 or BC1 complex. So this complex has the function of bridging the electron flow from photosystem 2 to photosystem 1. And at the same time, the problem of 2 electron transfer or the switching of 2 electron transfer from 2 electron transfer to 1 electron transfer is solved. And the second electron, which is directed into another pathway, it does not get lost. It arrives here at this station and then is taken up by another quinone, and this later on arrives later on again here in this station. So this process is also interesting because there are the 2 processes are linked with a gain of energy and a loss of energy. So electrons that are directed into this pathway, they confer a gain of energy, and electrons that are directed via this pathway, they confer a loss of energy. And the interesting point is that the amount of energy is here nearly identical. So, and Zhang comments on this. Remarkably, bifurcating electron transfer proteins typically send 1 electron uphill and 1 electron downhill by similar energies, such that the overall reaction is spontaneous but not profligate. So again, this requires highly precise parameters within this protein. And another interesting aspect about this is that the transfer of electron here is happens by means of quantum tunneling processes within the protein. And these processes are sensitive towards 4 parameters, and they are exponentially sensitive. 2 of them are the distance between the individual stations of the electron and the free Gibbs energy of the electron transfer. That means slight deviations from the parameters that are given would lead to the whole thing ceasing functioning. So there is absolutely no scope for evolutionary experiments. So selection and mutation will not work here, because even modest changes in the structure and of these proteins would simply hamper the whole construct functioning. So this is the anoxygenic photosynthesis apparatus. As we can see here already, it is rather simple compared to the oxygenic photosynthesis apparatus. It is composed of only 3 components. This is a photosystem. It can be either of type 1 or of type 2. And this is the complex 3, which has a similar function as for oxygenic photosynthesis. And this is an ATP synthase. So there is no second photosystem as in oxygenic photosynthesis. and electrons are withdrawn from other molecules than water. And overall, the electron flow is cyclic. So even if there was only one electron that perpetually cycled around these components, it would transfer energy. But the electrons are also used for carbon dioxide fixation. That is why they have to be withdrawn from other molecules into the cycle. So let's compare both apparatuses. In the oxygenic photosynthesis, we have 5 compounds with a linear electron flow, and there is an oxygen evolving complex. And in the unoxygenic photosynthesis, there we have 3 components and a cyclic electron flow. So the question is whether it is plausible here to assume that this can be that this can happen by evolution. So that we can go from unoxygenic photosynthesis to oxygenic photosynthesis by means of evolution. So the requirement here is to obtain or to add to this system the oxygen evolving complex and the photosystem 1, and to integrate them into the flow of electrons. So let's have a closer look to photosystem 1. So this is a crystal structure of of a photosystem from Heliobacterium modesticalium. So this is a prokaryote, and this is a rather simple type of a photosystem. So there are more complex ones, but this is suited well to serve as an illustration. So the weight of this photosystem is roughly 200 kilodaltons, and there are 60 prosthetic groups. That means molecules that serve as, for example, chromophores. And the chromophores within this complex are very densely packed. The concentration is huge. The concentration of the chromophores is about 0.5 molar. And for example, for keratin, it's 0.15 molar. So these are huge concentrations. And the dense packing of these pigments produces electrodynamic couplings between pigments. These couplings are highly sensitive to both molecular structure, which determines the pathways, and the mechanisms by which energy is transferred from an initial absorption to the target molecule. Again, there is absolutely no room for evolutionary trial and error. And excitation transfer occurs as fast as 100 femtoseconds. That means from one chromophore to the other within the light harvesting complex, which represents the fastest process of a single energy transfer step between 2 chlorophylls. And trapping the energy in the reaction center takes roughly 60 to 90 picoseconds. So this is the photosystem 1, and it is considered to be a rather deep energy trap. That means this is a funnel that is rather steep. Now, the difference in absorption maxima in the periphery and those in the center is rather large. So this is the view from the top. I just turn it around, and here in this red region you can find the reaction center. So the reaction center is very small compared to the light harvesting complex that is coated around it. So Nelson and Junge comment on this complex that the building of photosystem 1 involves a very complicated biogenesis and assembly process that brings all the photosystem 1 components together to generate the most efficient photochemical machine in nature. So let's have a closer look to the reaction center. So this small part here, yeah, it is also possible to work it out and to obtain such a structure. So we can see the components of the reaction center, where the excitation of an electron takes place. So the distance between the components here is a few angstroms. So this is very, very close. And this is absolutely necessary to enable quantum tunneling transfers, which happen in picoseconds. So here already electrons are transferred, not excitonic energy. And the geometrical arrangement of the cofactors is optimal. The quantum efficiency of this process is nearly 100%. So this is also interesting for us when we consider how to deal with the light of God. We should also have a quantum efficiency of nearly 100%. So Nelson and Yoga comment on this. Several genetic, biochemical and spectroscopic studies have shown that numerous cofactors are important not only for the function of photosystem 1 but also for its structural integrity. So apparently the proteins that hold together these cofactors serve as a scaffold that keeps the pigments in the correct geometry. So the whole thing has to have a certain structure, and it doesn't tolerate any small deviation. Also, the protein must play a role in controlling the energy levels of the pigments. Changes in conformation can strongly impact the electron transfer processes. There is a bacterium known which can even use this kind of conformation changes. In case the light intensity is very high, the photosystem can change its conformation so that it reacts to the light intensity. So the components we see here in this reaction center are chlorophylls, plastoquinones, and then there are iron-sulfur clusters. So this is characteristic of reaction centers of type 1. Let's have a closer look to photosystem 2. So here I have also drawn a red circle, and if you look very hard you can discover the oxygen evolving complex there. It's perhaps a bit difficult to see, but it's present there. I have checked. So this is a rather shallow energy trap. That means trapping the energy. So the funnel is not as steep as for photosystem 1. And this complex here is rather large. It has the weight of 350 kilodaltons. It is composed of 20 protein subunits, 95 chlorophylls, and 30 carotenoids. So this is quite a huge amount of molecules, which are all arranged in a highly ordered manner. Let's have a closer look to the reaction center. Similarly as for photosystem 1, again we have distances between the components of the reaction center of a few angstroms. And the quantum tunneling transfers again happen in picoseconds, within picoseconds. And the components are arranged in a highly optimal manner. And also here the quantum efficiency is about 100%. So there were also some experiments to measure the velocity of electrons being transferred from one component to the other. And this transfer here from the special pair in the middle, this is called special pair here, P680 means it absorbs light at the maximum wave, or the absorption maximum lies at the wavelength of 680 nanometers. So in the electron transfer here takes 3 to 4 picoseconds. And the second one, so from the phaophytin to the quinone, occurs at 200 picoseconds. And then it was also measured, this back transfer. And this takes around 100 milliseconds. So this is very slow. Why is that so? This is because after the charge separation, there is a strong tendency, there is a strong thermodynamic tendency for the electron to recombine. That means to return to the special pair. But this, of course, would render the process ineffective. And this is why these processes are very quick. They are very rapid. And this process is very slow. This means, again, the whole structure is highly teleological. It is constructed in a way so the electron will not return to the special pair. It is constructed in a way that the quantum efficiency is at maximum. So charge separation is stabilized in reaction centers in order to obtain maximum quantum yields. Again, this points to a teleological construction because a loss in efficiency is prevented. Another interesting aspect is energetics. So that means the excitation happens in a certain range of energy. And the photosystems occur in very different organisms. This is not a main aspect I will discuss here, but it is known nowadays that a common ancestry cannot be reconstructed. If you try to reconstruct from all the phototrophic organisms, if you try to create some kind of phylogenetic tree, you will not succeed. But what I'm interested in is the feasibility of the claimed evolutionary conversions. So that means from the unoxygenic photosynthesis apparatus, which is rather simple, to the rather complex oxygenic photosynthesis apparatus. Again, what we can see here, in the oxygenic photosynthesis, electron transfer is rather linear, or rather, it is linear. And here in unoxygenic photosynthesis, the electron transfer is circular. And even more important is that here we have a different redox potential of the whole photosystem. So the photosystem 2 here has a much higher redox potential than the photosystem 2 of many phototrophic prokaryotes. So if it is claimed that there was an evolution from unoxygenic photosynthesis to oxygenic photosynthesis, the whole redox potential of a photosynthesis must have been changed during evolution. So all compounds had to be changed in their redox potential to obtain oxygenic photosynthesis. And this has to happen in a concerted manner, which is impossible for evolution. You can also compare it roughly to a running engine that you try to rebuild screw by screw. So let's come back to this picture here, the comparison of oxygenic photosynthesis and anoxygenic photosynthesis. What would be required to have an evolutionary transition from anoxygenic photosynthesis to oxygenic photosynthesis? We'd have to integrate the second photosystem into the whole thing and couple the electron flow. And at the same time also the oxygen evolving complex. And then there has to be a change from the cyclic electron flow to a linear electron flow. And the potential of all components of photosystem 2, this is what I mentioned before, the electrochemical potential has to be shifted completely in a concerted manner. Simply put, this seems to be completely impossible by means of evolution. So let's come to rhodopsin photosynthesis. This is the third type. So far I have discussed two types. The oxygenic photosynthesis with chlorophyll chromophores and anoxygenic photosynthesis with chlorophyll chromophores. And this is a totally different construct, the rhodopsin photosynthesis. Again, this is a crystal structure. It is roughly barrel-shaped, and here in the middle there is retinol. There is a retinol molecule, and this retinol molecule it is also transformed by light from trans to cis. There is a double bond, and it switches from trans to cis whenever there is a photon arriving. And to make this visible, I have manipulated the crystal structure so we can have a better look on the retinal. So this is a full space depiction, and it is coated by residual parts of amino acids in the protein. So what we can see is this retinal is set under pressure by the space-filling molecules here, which are tryptophans. And whenever a transition from trans to cis occurs as a consequence of a photon that arrives at the retinal, there is strain that is built up as a result of the isomerization. And the conformation of the whole protein changes. So there is pressure exerted on the retinal to return to the trans conformation. And this process leads to a constant isomerization of this molecule. And this is linked to a proton flow. So there is a proton arriving at the nitrogen atom of retinal, and it is further directed via a chain of amino acids from one side to the other. And this is linked to the isomerization cis and trans. And the isomerization happens at a time scale of femtoseconds. So this is the light falling on the retinal. Another interesting aspect is the bacterial retinal and the one of humans and animals. The isomerization happens at another double bond in bacterial retinals than in the one of humans and animals. And also, the isomerization happens here from trans to cis, and it returns spontaneously, which it does not in humans and animals. Here, the photons cause a relaxation, an energetic relaxation from the cis retinal to the trans retinal, and an enzyme performs the return to the cis conformation, to the cis configuration. So Kandori comments on this aspect with the following words: microbial and animal rhodopsins share a common architecture of 7 transmembrane alpha helices with the N and C termini located extracellularly and intracellularly, respectively, but have almost no sequence homology and differ largely in their functions. So what they state is, though there is a similar molecule, the way it functions is totally different, and the proteins have no sequence homology in bacteria and humans and animals.
Is that true for the epoxy?
It's also for the chlorophyll is also the same. The sequence homology of the two photosystems is nearly 10%. So now I come to some conclusions. First of all, for the chromophores. The chromophores have extremely high extinction values. They have the tendency to stabilize electronically excited states, and there is a great diversity in nature. And different kinds of animals have complementary chromophores in different environments. So they are complementary. Blankenship stresses that in contrast to the reaction center complexes, antenna complexes are remarkably diverse, strongly suggesting that they have been invented multiple times during the course of evolution to adapt organisms to widely varied photic environments. So what we see is the statements fits very well to creation. They have been invited independently, but it does not fit very well to evolution. And when it comes to designing artificial light harvesting systems, we can learn a lot from, they say always, nature, but of course this is a replacement for God. This is also a citation from a scientist. The aim of this review is to condense what has been learned from studies of natural light harvesting complexes into a few important lessons that can aid the design of synthetic systems. So then there are also proposals how it might have happened. Of course, there is always this pattern that we go from the simple organism to the most complex. So the narrative is that we start from a non-phototrophic organism, going via an anoxygenic photosynthesis, and then arriving at oxygenic photosynthesis. So this is the basic story. And then there are two different models. There is the model of the selective loss. So starting from an organism that possesses no photosystem, a photosystem is obtained by some kind of evolutionary transition. And it doesn't produce oxygen so far. So this is an anoxygenic phototroph. And then there is a gene duplication, and this organism still does not produce oxygen. And then, by mutation and selection, different organisms form. So this is the selective loss. This is a complex ancestor, and starting from this complex ancestor, different kinds of organisms should have formed. However, this kind of transition has never been observed in nature, so this is not known that something like that can happen. And also, such an organism that contains a photosynthesis apparatus with two kinds of photosystems that are quite similar is not known. Yeah? Also, this transition from C to E is extremely demanding, and it would mean that there must have been a changement, as I already stated, of the electrochemical potential of a whole photosystem. So this is the fusion model. In the fusion model, it is assumed that starting from an organism where there is no photosystem, a photosystem is obtained, and then two different kinds of photosystems formed, type 1 and type 2, and then they are combined by lateral gene transfer to an organism, which then can produce oxygen. However, this has never been observed in nature again, and also the lateral gene transfer is completely inappropriate to transfer such a huge amount of genetic information to form a photosystem. And also the transition from C or D to E, that means obtaining two photosystems in one organism where there were one, this is extremely demanding, and this has also never been observed. Another problem is that the sequence homology between photosystems of type 1 and photosystems of type 2 is about 10%. So nearly no sequence homology. And there are also comments on this by several scientists who are somewhat astonished by this fact. Although at the sequence level, there is almost no detectable similarity between reaction center 1 and reaction center 2. The structural and cofactor arrangements are unquestionably homologous and clearly indicate common ancestry. So this is somehow contradictory. Or Nelson and Junge say the explanation for this, that means that these different sequences of photosystem 1 and photosystem 2, the explanation for this may be hidden in the protein structure that, at least in the core, was highly conserved over more than 3.5 billion years of evolution. So it has never changed. But this is not surprising, because it cannot change. It will cease functioning if it changes. Approximately 90 chlorophylls have remained largely unchanged by the more than 2 billion years of evolution between cyanobacterium and flowering plants. So I do not want to say that I believe that there were several billion years of evolution, but what the scientists say is that there was hardly any change in these photosystems, be it the reaction center or also the light harvesting complexes. There was no change. Or Rosenberg, despite this similarity, microbial and animal rhodopsins share no detectable sequence homology. This peculiarity has been attributed either to a deep divergence between the evolutionary related families of transmembrane proteins or to convergent evolution. So the data bespeak unambiguously that evolution never took place, because photosynthesis apparatuses are said to be conserved for over 3 billion years. This means even proponents of evolution admit that they never changed. At the same time, they are highly diverse. So if diversity is obviously not the result of evolution, the only reasonable explanation is created diversity. So I come to the conclusion: photosynthesis apparatuses are highly teleological entities. They also require a highly efficient photoprotection, which is even more sophisticated than the photosynthesis itself. Photosynthesis apparatuses are highly diverse, and no subject of evolutionary changes. Evolutionary models fail constantly and completely to explain the origin of photosynthesis apparatuses. So it is required that there is photoprotection and a complex photosynthesis apparatus at the same time. Both have to be formed at the same time, otherwise it will not function. Overall, the present analysis makes it clear that the structural exactness and functional precision of the three types of photosynthesis apparatus, rhodopsin, chlorophyll, anoxygenic and oxygenic, are opposed to models of an evolutionary explanation. Furthermore, it has often been noted that the rich diversity of the subgroups of the two types does not allow the construction of a meaningful evolutionary genealogy. Rather, the nature of these masterpieces of molecular architecture speaks to the great genius of the creator, who produced them to enable living organisms to occupy many ecological niches. So, to him be the glory. I thank you for your attention, and I look forward to the questions.
So, thanks a lot, Boris, for this very interesting talk about the extreme fine-tuning and irreducible complexity of life's orbiting systems. So, I think we have time for one or two questions. So, Pekka, please.
Thank you very much, Boris, for killing evolution. But one point, they teach us that there was no oxygen on this planet, so no ozone layer. What's your point that could this anaerobic photosynthesis have developed without the protection of the ozone layer?
No, I don't think so, because...
Please repeat the question.
Okay. The question was that it is assumed that there was an atmosphere at the beginning, an atmosphere which was devoid of oxygen. And in this atmosphere devoid of oxygen, the first organisms, the first phototrophic organisms should have formed. Is that plausible? And I can only say, well, an atmosphere that is devoid of oxygen is not able to form ozone. There is no ozone layer, so this will not protect the organisms from UV irradiation. And UV irradiation will kill the organisms. So either they hide in some regions that are far under the ground, where no light will reach them, but then they cannot be phototrophic, or they creep on top and try to start photosynthesis, and will quickly die. So these are the two options, and I don't know how it is solved. So as far as today, well, I have studied the theories of the great oxygen event, and it is not plausible at all. The second point is also that oxygen is poisonous, so it is toxic for the organisms, even in very small amounts. So the first organism that produced oxygen should have killed himself.
Yes,
I was just wondering, for this question, these organisms that consume light for energy, are they the only ones that would produce oxygen, or are there other candidates for that process? If there was an environment without oxygen, are these organisms that are light consuming the only ones who would produce oxygen, or are there other candidates? You mentioned in the first page that there were several ways of consuming energy, which you would only go through these ones, but there were organisms taking energy from stones or other parts. Would any of those be producing oxygen? Or is this the only
No. So the question is whether heterotrophic or lithotrophic organisms that have no capability of photosynthesis could produce oxygen.
and then transform
Yeah.
into this.
The production of oxygen is only possible if photosynthesis is present. An organism is not able to produce oxygen without a photosynthesis apparatus. There are organisms that can shift from, for example, aerobic respiration to anaerobic photosynthesis. There are some microorganisms that can do that, but they have to have this information for photosynthesis to produce oxygen.